Welcome everyone, Dr. Matson again. We're going to go over research methods of biopsychology and try to understand a little bit better about what biopsychologists do. So, to take an X-ray photograph of an object, we have a beam of X-rays that's passed through onto a photographic plate, and any part of that object that absorbs X-rays differently than the surrounding medium will be distinguishable. But standard X-rays can't be used to study the brain because it's composed of too many overlapping structures that all absorb X-rays to about the same degree. But the contrast X-rays solve this problem. And so we look at other material that's introduced into the structure to make it kind of stand out from others on an X-ray. So if we're going to do like a cerebral angiography, we're going to inject a dye into the carotid artery, and it's going to reveal the displacement of enlargement of blood vessels is what we'd be looking at. And so the X-ray CT provides a three-dimensional view of the brain, and the X-ray gun and the X-ray detector rotate in apposition around the brain at one level, taking a series of measurements from which an image of one section is constructed. And so this is then repeated at multiple levels. And so the CT scan image of the brain is not considered to be high resolution with this, though. So this just shows you the illustrations, of a scanning, shows a person lying on a patient trolley. We place the head in the hole of the huge donut-shaped structure that rotates clockwise. On one side of the interior of the structure is an X-ray source, and on the side exactly opposite to that is the X-ray detector. So this rotating structure is housed, in this big tube that, we essentially slide you into. The output is shown as a series of horizontal CT scans, which is white and gray images on black backgrounds. And then you can see the person seated at the computer is viewing these three-dimensional reconstructions of the scans. The images of the brain shows in three perspectives that we see on the monitor. So the pair of PET scans here, is a scan that was done when the volunteers' eyes were either open left or closed on the right. All right, so open eyes on the left, closed eyes on the right. Areas of high activity are indicated by red and yellow. So you want to notice the high level of activity in the visual cortex of the occipital lobe when the volunteers' eyes were open. The images are similar, but the major difference is the occipital lobe, and that's what we see highlighted in the left image in red and yellow, and then green in the right image. So MRIs are the imaging that we do that offers a higher resolution than what the CT is going to offer. And so the images are measurements of the waves emitted by hydrogen atoms, and then they're placed in a magnetic field. Its high spatial resolution stems from the fact that neural structures differ considerably in their density of the hydrogen atoms. The diffusion tensor imaging, DTI, provides a means of imaging the axonal tracks rather than the cell bodies. The technique is based on the rapid diffusion of water molecules along tracks. DTI is unique, and so in the way that it allows investigation of interconnectivity among structures, and some connections of simple organisms have been completed. The functional MRI, or fMRI as it's referred to, measures the increase in oxygen or blood flow to all active areas of the brain. And so the four advantages the fMRI has over the PET include: you don't have to inject anything into the subject. One image provides structural and functional formation. The spatial resolution is better, and we can also measure changes in real time. The disadvantage to the fMRI is that the images are not of neural activity, as many assume. The signal recorded from an fMRI is called the BOLD signal, which is the blood oxygen level dependent signal. And these fMRI scans are too slow. They take 2 or 3 seconds compared to many neural responses, which can occur in milliseconds. The figure here shows the colored MRI scan of the head. The brain is highlighted in blue, it's got patches of yellow, and then the rest of the head is highlighted in yellow and red. So this is a color enhanced MRI. In this image, we can see that the head shows the upper left portion of the head that's cut away, and it shows us a tumor, and this is colored in red. And this is an image of the diffusion tensor MRI. And so this shows the lateral view of the brain, which is gray against a black background. And then we've got small patches of the cortex that are highlighted in red, orange, and black. This image shows the lateral view of a brain, which is gray against a black backdrop. And then it shows small patches of the cortex that are highlighted in red, orange, and black. Doing a functional ultrasound imaging, or FUS, is a new imaging technique that uses ultrasound, which is sound waves of a higher frequency than what we can hear, to measure changes in blood volume, especially in particular to certain brain regions. When a brain region becomes active, blood levels increase there and alter the passage of ultrasound through that brain region. The transcranial magnetic stimulation, or TMS, allows researchers to non-invasively stimulate the brain using a focused magnetic field to disrupt brain activity and produce a temporary lesion. The effects of disruption on cognition and behavior are then assessed. With the TES, the transcranial electrical stimulation, this is a technique that's used to stimulate or turn on an area of the cortex by applying an electrical current through 2 electrodes placed directly on the scalp. And then the last one, the transcranial direct current stimulation, TDCS, can be used to temporarily stimulate an area of cortex while observing the effects on cognition and behavior. The scalp EEG, this is a signal that's measured through an array of scalp electrodes. The EEG waves reflect the sum total of all of the electrical events in the head, like back to the last chapter with the APS, the EPSPS, the IPSPS, also eye movements, blood flow. The EEG reveals little about the nature of the underlying neural activity. Its value, though, lies in the fact that particular EEG waveforms are associated with particular states of consciousness. Generally, low amplitude or fast EEG activity is associated with an alert and aroused state. So high amplitude or slow EEG activity, alpha waves, is associated with a relaxed but awake state. So EEG can be used to study brain activity in real time in response to specific events. These are called evoked potentials. Usually many evoked potentials are used to generate an averaged evoked potential in order to reduce the noise of the background EEG activity. The EEG recordings are a valuable diagnostic tool. So somebody like the presence of a high amplitude spikes in the EEG, this would be the primary criterion for diagnosing epilepsy, right? If we see the high amplitude spikes. Epileptic spikes is what we refer to them as. The MEG, this brain activity is measured in terms of changes in the magnetic fields on the surface of the scalp. So when we look at the different types of waves here, the aroused one is a series of waves with very short amplitude and high frequency, almost resembles a straight line made of kind of the minute zigzags. The relaxed is the alpha waves. These are the alternate with waves of lesser amplitude and higher frequency at regular intervals. The alpha waves are a series of 5 to 7 waves with high amplitude and then low frequency occurring in succession. The asleep, this is the almost an irregular straight line with periods of waves with a very short amplitude. And then deep sleep resembles a highly irregular sine wave without a fixed baseline. So the image here shows 10 segments of 2 cases. So 1 second segments of background EEG. And then we have 10 segments consisting of waves that are of very small amplitude that do not deviate much from the baseline. The average background EEG is a slightly irregular straight line. 1 second segments of EEG with evoked potential. 10 segments consisting of waves that are very small amplitude but that deviate highly from the baseline. The average evoked potential is shown after a click. So it starts with a series of small waves followed by a bigger wave, which then returns to the baseline. The line graph shows the average auditory evoked potential plotted on an axis with time in milliseconds, ranging from 0 to 600, plotted on the x-axis. After the meaningful click at 0 milliseconds, there's a series of small waves labeled as far-field potentials. The line then slopes downward with small upward peaks. The lowest downward peak is reached at about 300 milliseconds, after which it goes upward and reaches the baseline at around 550 milliseconds. This just shows a picture of a woman patient. She's under the MEG machine for observation. So for muscle tension, we're looking at the EMG, and this measures the changing difference in the voltage between two large electrodes placed on the skin above a large muscle. The amplitude of the EMG signals indicates that the combined level of tension in the underlying muscle. The raw signals are usually integrated so that the data is easier to work with. So the height of the curve of integrated EMG activity indicates the number of spikes in the EMG signal per unit of time. And for the eye movement, the EOG, eye movements are recorded by placing 4 electrodes around the eye. The signal results from the fact that the front of the eye is more positively charged than the back. And the direction of movement can be inferred from the relation between the activity recorded on 2 channels, like above versus below or left versus right, something like that. The raw EMG signal appears as a series of waves with such high frequency that it appears as a thick red blot, essentially. The amplitude of the waves is much higher initially and then tapers. And then the corresponding integrated EMG shows a signal with a line with an initial upward slope, which then slopes downward and then kind of plateaus out. This image shows the 2 graphs, one plotted using the electrodes connected above and below a woman's eye, and another plotted using electrodes placed to the right and the left of the eye. But both graphs show waves with peaks and troughs, with the peaks of one graph almost corresponding to the troughs of the other graph. The SCL, or the skin conductance level, is the general level of skin conductance associated with a particular situation. And so the skin conductance response is a rapid change in skin conductance to a particular event. So one application could be the lie detector test. The cardiovascular activity would be the ECG, and this shows changes in cardiac function, including heart rate, which reflects changes in emotion. Blood pressure is expressed as a peak pressure during systoles over minimum pressure using the diastoles. 130 over 70 is normal. 150 over 90, or greater than 150 over 90 is considered hypertension. The device is commonly used to measure blood pressure. And then the measure of the blood volume, this is done either with a strain gauge or by measuring the amount of light absorbed by the structure. If we're going to measure the volume of blood in a body structure, we might do this using a penis. The first step in many invasive biopsychology experiments is the stereotaxic surgery. It allows accurate placement of lesions, probes, electrodes, and other devices into the brain. And the reference point is often the bregma. This is the point where 2 main plates of the rat skull come together. This is just a photo of the stereotaxic instrument that could be used. It's meant for surgery on rodents. So the three images that we see here, we have the rectangular atlas that shows the cross-section of a rat's brain with the amygdala highlighted. The atlas is graded on all four sides from 0 to 10, top to bottom, on the left and right edges, and from 7 to 0, and then to 7, that is 15 markings on the top and the bottom. A black dot on the amygdala corresponds to 4.5 on the scale at the bottom, and 8.5 on the scale to the right. Above the cross-sectional image is the label bregma 2.8. And so it states that the stereotaxic atlas indicates that the amygdala target site is 5 millimeters ventral. This page of the atlas represents a frontal section that is 2.8 millimeters posterior to the bregma. The second image shows a rat being held in place, and then an electrode holder is being inserted into its head. A hole is drilled 2.8 millimeters posterior to the bregma and 4.5 millimeters lateral to it. Then the electrode holder is positioned over the hole, and the electrode is lowered 8.5 millimeters through the hole. The third one is a rat with an electrode and electrode connector at its head, and it states that the electrode is anchored to the skull with several stainless steel screws and the dental acrylic that is allowed to harden around the electrode connector. The electrode is seen as a sharp pin with the electrode connector at its tip. So when we look at invasive physiological research methods, the lesion method is what we see here. Aspiration method is often used to remove cortical tissue. The radiofrequency or high frequency is the electrolytic lesion. It's the most common subcortical lesion. The tissue is destroyed by the heat of the current. Small knife cuts are often used for severing tracts. And with the reversible lesions, these are useful because the same subjects can be repeatedly tested. Tissue can be cooled to the point that all neural activity temporarily stops. Lesion studies must be interpreted with caution. A lesion inevitably damaged structures other than the one that its surgeon targeted, and lesions seldom remove a targeted structure completely. The behavioral effects of unilateral lesions are much milder than bilateral lesions. In this image, we see a thin rod-like structure inserted into the brain, a blade attached to its tip, which is at approximately 45 degrees to the rod and points upward, and then is seen near the anterior commissure. The effects of electrical stimulation are often opposite to those of a lesion to the same brain site, and electrical stimulation research is done prior to any lesioning that would occur. The intracellular unit recording, this method changes in the membrane potential of a neuron over time. It does require a microelectrode positioned inside a neuron. It's next to impossible to record the intracellularly in a freely moving animal because it's difficult to keep the microelectrode inside the neuron. If we're looking at the electrocellular unit recording, in this method, a microelectrode is positioned near a neuron. The signal is a series of spikes, and each spike indicates an action potential from a nearby neuron. Spikes of the same amplitude are assumed to come from the same neuron. When we look at multiple unit recording, this type of recording provides an indication of the rate of firing of many neurons in the general vicinity of the electrode tip. An electrode larger than a microelectrode picks up an action potential from many nearby neurons. The signal is then integrated so that the height of the curve indicates the number of action potentials in the vicinity per unit of time. And then the invasive EEG recording, this one, implants electrodes are used to record EEG in laboratory animals because the scalp electrodes do not allow as clear or finite position recording. So the various routes here of drug administration, the peripheral routes, all of these potential routes listed, all suffer from the fact that many drugs cannot pass the blood-brain barrier. And this problem can be overcome by administering drugs via intraventricular or by a microinjection of drugs directly into the brain tissue. Sometimes drugs can be injected into the brain that produce chemical lesions that are more selective than electrical lesions might be. For example, the 6-OHDA is a neurotoxin that selectively destroys the dopamine and noradrenergic neurons in the vicinity of the injection site. Radioactive 2-DG is injected, and then the animal performs the behavior of interest. Then the animal is immediately killed. Its brain is removed and sliced, and the slices are coated with a photographic emulsion. And after a few days in the dark, the areas of the brain that were particularly active during the test show up as dark spots of radioactivity. A technique that allows the measurement of specific neurotransmitters in behaving animals is called the cerebral dialysis. And this is a fine U-shaped tube that's inserted into the brain of an animal. Its tip is made of the semi-permeable dialysis membrane, which is a solution of artificial cerebral fluid that circulates through the tube. Because the tip of the tube is semi-permeable, chemicals can continuously move from the brain into the solution in the tube. It goes down a concentration gradient, which can then be removed for later analysis using a chromatograph. So this image shows the 3 frontal sections that are in different colors against a black background. The color coding next to it indicates 941.23 NCI per G for red, which decreases for each color of the spectrum, and then ends at 0.16 for violet. In the sections in the right hemisphere, the lateral posterior and dorsal lateral and superior and the visual cortex appear as bright yellow patches. Immunocytochemistry begins by injecting antigens or foreign proteins into an animal so that the animal will create and bind antibodies to the antigen to remove or destroy it. Antibodies for the most of the brain's peptide neurotransmitters and receptors have been created. These can be labeled with a dye or some kind of radioactive element and then used to identify specific neuroproteins in slices of brain tissue. The in situ hybridization allows peptides and proteins in the brain to be located, labeled hybrid RNA strands with the base sequence complementary to the mRNA for synthesizing the target neuroprotein. The hybrid RNA binds to the complementary mRNA in the target cells and allows the target neuroprotein's location to be marked. This is just a photo that shows the rat's pons after injecting an antibody into the rat's body. These techniques involve the creation of organisms that lack a specific gene, and any measurable neural or behavioral anomalies are then noted. So an example of genetic engineering would be the melanopsin knockout mice. The melanopsin is a protein found in some neurons in the mammal's retina, which has been implicated in the control of the circadian rhythms, since many of the neurons project to the hypothalamus. It was discovered that the knockout of the gene reduced but did not eliminate the responses of the clock to changes in light. The melanopsin contributes to the control of the circadian clock, but it's not the only factor. The problem is that most behaviors are determined by multiple genes. So eliminating one gene usually alters the expression of other genes, and then gene expression is dependent upon experience, which again might be altered by the absence of the missing gene. So these techniques involve the replacement of one gene with another, and sometimes genetic information from a different species is implanted, creating a transgenic subject. In some applications, pathological human genes are used to create transgenic mice, which are then used to study the neural basis of human disease. But in other applications, genes are manipulated so that researchers can turn them on or off with light or chemical sounds, or signals rather, sorry. So this gene knock-in technique, it's possible to replace one gene with another or add a gene that doesn't exist in an organism. Such gene knock-in techniques have created interesting possibilities for both research and therapy. As a means of controlling for the interaction of genes with experience, many researchers now use modern gene editing techniques. These new gene editing techniques allow researchers to edit genes at a particular time during development. The CRISPR, or Cas9 method, is generating a great deal of excitement in the scientific community, and we will talk more about this in class. The GFP gene can be inserted into target cells that are later exposed to blue light. This produces bright green fluorescence for visualization. The GFP gene can be altered to synthesize proteins that the fluorescence in different colors show. This is just a photo that shows touch receptor neurons that are labeled by proteins in the fluorescent green. This photo shows the neural axon, which is traced in different colors. Opsin genes are inserted into a particular type of neuron, and scientists can use the light to hyperpolarize or to depolarize cells. So there's kind of a modern approach to neuropsychological testing. Traditionally, a single neuropsychological test was administered to a patient, but this approach changed in the 1960s to include a standard battery of neuropsychological tests. And currently, it's in its most common form for a customized test battery approach to be used instead of the single neuropsychological test or the standard battery. The WACE intelligence test was the most neuropsychological assessment. It begins with the Wechsler Adult Intelligence Scale. It has 11 subtests, 6 compromise, the verbal scale, 5 compromise for the performance scale. Then we have the memory or the information subtest and the digit span test. These are memory tests that compromise part of the WACE. However, they're notoriously bad measures of memory. The language, when we look at language, the WACE verbal subsets reveal significant language impairments in a variety of ways. So if the WACE was not done, the token test is a good initial screening for test when we look at language-related deficits. There's 20 tokens of 2 different shapes, 2 different sizes, and 5 different colors. The subject is asked to carry out various acts such as touch the small blue circle, and then the large green square. And if the token test identifies language deficits, then it's followed by a battery of tests of language ability. If memory deficits are discovered, then the neuropsychological attempts to answer 4 key questions. Is short-term or long-term memory affected, or both? Are the deficits anterograde or retrograde, or both? Do the deficits involve semantic or episodic memory, or both? And then do deficits involve explicit memory or implicit memory, or both? And so patients with speech problems must be further tested for problems of phonology, syntax, or semantics. Patients with dyslexia must be tested for reading or pronunciation problems for language. In addition to being interested in which brain regions are active during particular cognitive tasks, cognitive neuroscientists are also eager to understand how network activity across multiple brain regions is related to a particular cognitive task. This approach is referred to as a study of functional connectivity. To measure functional connectivity, a cognitive neuroscientist examines which brain regions have parallel patterns of activity over time. So these behaviors are displayed in the same form by virtually all members of a species of the same sex, like grooming, swimming, nest building. The open field test provides 3 measures of emotionality. We have the degree of inactivity, the thigmotaxis, and the defecation. And then for aggression and defense, we can study those by recording encounters between a small male intruder and a colony's dominant or alpha male. Aggression involves a sideways approach, sideways pushing, biting directed at the back, Defense involves boxing, rolling over onto the back, biting the face, freezing, as well as fleeing. For the rat's sexual behavior, this is another species that common behavior that's widely studied. We look at two common measures of female rat sexual receptivity. And then the male rat behaviors include number of mounts to intromission, number of intromissions to ejaculation, and time to reinitiate mounting after the ejaculation, or what we would call the post-ejaculatory interval. Looking at traditional conditioning paradigms, this plays an important role in biopsychology for three reasons. Conditioning is a phenomenon of primary interest to psychologists. Conditioning procedures are often used to train laboratory animals to perform as required in behavioral experiments, and researchers can infer much about the psychological state of an animal from its ability to learn and perform in various responses. And so with Pavlov, we see the UCR, UCS, CR, and CS. I got to put these in a different order. The UCS is the unconditioned stimulus, and that is followed by the unconditioned response. The conditioned stimulus then follows with the conditioned response. And so with Pavlov's experiment, we see the salivating dog, and so the UCS would be the food. It's unconditioned. We show food to a dog. We don't have to train. It's just something naturally that they want. And so the UCR would be the salivating, the drooling. So we show a dog food, they naturally salivate. Unconditioned stimulus, unconditioned response, no training, no nothing, just natural. And then we pair the food with the bell. So we ring the bell, food, over and over again. And the dog is starting to associate, right? Pavlov conditioning, all about associative learning. The food and the bell together. And so the dog continues to salivate. And then all of a sudden we pull the food and just ring the bell. The bell becomes the conditioned stimulus. And the dog continues to salivate, the conditioned response. So with the UCR and the CR, we always want the same response, just from two different stimuli. And then Skinner is operant conditioning. And with operant conditioning, we look at reinforcement and punishment, positive reinforcement, negative reinforcement, positive punishment, negative punishment. Positive reinforcement or negative reinforcement, it doesn't matter if it's positive or negative. Reinforcement, we always want behavior to continue. Punishment, we always want behavior to stop. So we either add or take away from a situation to make that happen. The taste aversion conditioning experiments that were done in the 1960s challenged widely held views of learning that had outgrown the study of the traditional conditioning paradigms. And so the view that learning is a gradual step-by-step process by showing that it could occur reliably in one trial. And also the view that temporal contiguity is necessary for learning by showing that conditioning occurred even when the taste and distress were separated by several hours. And then the view that, the third one here is the view that associations between two stimuli are equally easy to learn by showing that rats could learn the relation between the gastral distress and a taste, but not a light. And so this is with the conditioned taste aversion trial that was done. With the radial arm maze, this was used to study foraging behavior in the laboratory. And foraging in the wild is complex. So the rat must learn where food is likely to be, but not to immediately revisit a stripped site. In the radial arm maze, rats quickly learn to go directly to the arms that are baited with food each day, but they rarely visit the same arm twice on a given trial. For the Morris water maze, this is another laboratory paradigm that was used to study rat spatial ability. And the Morris water maze is a large tub of milky water. To get out of the water, rats must learn to swim a slightly submerged invisible goal platform. They use external room cues to solve the location. And the last one here, the conditioned defense burying. This is based on the observation that rats exposed to inanimate object that has been the source of a single aversive stimulus, like a shock or a bad odor, maybe an air blast, a flash, they'll often bury it. It's been used to study anti-anxiety drug effects, and anti-anxiety drugs reduce conditioned defensive bearing at low doses anyways. This image shows you a cylindrical structure with 8 arms at its top end. Each arm is the form of a bridge that connects to a small rectangular tray at its end. Some of these trays have food in them, and then the rat would be at the center of the cylinder. These photos show you the defensive bearing. So a rat's in an enclosure, a small rod is pointing from one wall of the enclosure, and then the rat is facing it from a distance. The floor is covered in a thick layer of a granular substance, and that's photo 1, which is the top left one. Photo 2, top right, the rat starts moving the granules. Photo 3, bottom left, it has covered half the rod with granules. And then photo 4 on the bottom right, it has covered the rod fully and is looking at the heap from a distance. And this brings us to the end of this PowerPoint. So multiple research methods almost always need to be used to answer a question. Seldom, if ever, is an important biopsychological issue resolved by using a single method. The reason for this is that neither the method used to manipulate the brain nor the methods used to assess the behavioral consequences of these manipulations are totally selective. There are no methods of manipulating the brain that change only a single aspect of brain function, and there are no measures of behavior that reflect only a single psychological process. So I hope this helps understand a little bit more about biopsychology and the research that we do. Thanks for listening, and reach out if you have questions.